Bismillahirrahmanirrahim. Assalamualaikum and hi everyone. In this video, we will be presenting our case study for analytical chemistry entitled Determination of Glucose Levels in Blood Using Paper Chromatography. We chose this topic because it relates to real-life application of analytical techniques, especially in medical field. The background of this study is that glucose is a type of sugar found in the blood, and it is the main source of energy for the body. Monitoring glucose levels is very important, especially for managing diabetes. In analytical chemistry, paper chromatography is a simple and cost-effective technique that is used to separate substance. This method can also help detect glucose from complex biological samples like blood. However, detecting and separating glucose from blood can be difficult because blood is a complex mixture. Some methods to test glucose require expensive and advanced instruments. Therefore, a simple and low-cost method like paper chromatography can be useful for this purpose. Our study has three main objectives. First, to understand how paper chromatography works as an analytical technique. Second, to explore how this method can be used to detect glucose in blood samples. And third, to identify the advantages of using paper chromatography in simple biological analysis. Through this case study, we aim to show how a basic chemistry technique can be applied to important medical situations in a simple way. Now, I will explain the methodology used in this case study. This case study is fully based on secondary data. All the information we gathered came from online sources such as articles, journals, and official websites. We collected the data by searching through platforms like Google, Google Scholar, and other scientific websites related to analytical chemistry. Keywords such as paper chromatography and glucose detection were used to help us find relevant sources. In selecting the data, we only used information that was reliable, relevant, and updated. Priority was given to sources that had scientific value and clear explanations. For the scope of our study, we focus only on the principle and application of paper chromatography, specifically for glucose detection in blood. We do not cover other types of chromatography or any hands-on lab experiment for this glucose itself. However, we have previously conducted a basic paper chromatography experiment using food coloring. Even though it wasn't related to glucose, that activity helped us understand how the separation process works on filter paper. This practical experience really helped us understand the technique better for this case study.
The theory of chromatography is based on the principle of separating components of a mixture by distributing them between a stationary phase and a mobile phase. The stationary phase is a solid or a liquid coated on a solid, while the mobile phase can be a liquid or a gas that carries the sample through the stationary phase. Separated molecules are components of the mixture that are separated based on their interactions with the two phases. This chromatography theory is important because it is essential for separating, identifying, and quantifying components in complex mixture across many fields. The theoretical models. The first model is plate model theory. It conceptualized the chromatographic column as divided into many discrete layers. Then each plate represents a site where the sample equilibrates between stationary phase and mobile phase. The second one is the rate theory. Rate theory focuses on the factors that causes band broadening, such as eddy diffusion, longitudinal diffusion, and resistance to mass transfer. The van Diameter equation relates plate height to mobile phase velocity and these broadening factors. Van Diameter equation. H stands for reduced plate height, a dimensionless measure of the band broadening. A, B, and C are constant, and V stands for reduced velocity. The process of determination of glucose levels in blood using paper chromatography works based on the theory. These are the short steps how the process works. Firstly, a small blood sample containing glucose is applied as a spot on chromatography paper. Then the paper acts as the stationary phase and a suitable solvent system as mobile phase is allowed to move through the paper by capillary action. After that, as the solvent moves, glucose separates from other blood components based on its affinity for the stationary phase and mobile phases. After separation, glucose is identified by its position RF value relative to standards. Lastly, a colorimetry agent such as anthrone is applied to develop a color spot corresponding to glucose. Next example of material and step. For the material, we need Wattman number 1 chromatography paper, capillary tube or micropipette, solvent and butanol acetic acid water in 4:1:5 ratio, glucose standard solution and blood serum sample, iodine vapor or other staining reagent, and lastly chromatography chamber. After that, for the step is 1, draw a pencil line 2 cm from the bottom of the chromatography paper. 2, spot the glucose standard and the blood sample on the line using a capillary tube. 3, allow spot to dry. 4, place the paper in a chromatography chamber with solvent below the line. 5, let the solvent run. 3, pour up the paper. 6, remove and dry the paper. 7, spray with staining reagent like Benedict reagent or iodine vapor to visualize the glucose. 8, measure and calculate RF value. This is for references for the step.
Example of the result, here the glucose standard travel 4.0 cm and the solvent move 10.0 cm, giving an RF value of 0.40. For the blood sample, the spot moved 3.9 cm, which is a very close, giving an RF of 0.39. So, this indicates the presence of the glucose in the blood sample. Next, we will continue about application of daily life based on our presentation. The first application is in education demonstration in labs and classroom. This method is used to separate sugar like glucose, fructose, and sucrose. It helps students understand the concept of separation and how RF values works. The second application is preliminary glucose detection in blood. This method detects the presence, not the exact quantity of the glucose in the dilute or treat blood samples. It is especially useful in basic research or setting without advanced instrument. Third, in the food industry, paper chromatography is commonly used to detect different types of sugars in the processed products like soft drinks, syrup, and candies. This method ensures accurate sugar contents, which is important for quality control and food safety. Fourth, in forensic science, it helps detect biological compounds such as sugar, drugs, or toxins in body fluids, blood, and urine. It is very useful in solving criminal cases, especially to identify causes of death or drug abuse. 5. In the pharmaceutical industry, paper chromatography is essential for checking the purity and composition of chemical ingredients in drug formulation. It helps scientists detect any unwanted substance and confirm that the correct compounds are present in the right amount, ensuring drug safety and quality. Lastly, in environmental and botanical studies, this technique is used to identify pigments like chlorophyll or anthocyanin in plants, as well as measure plant content in plant sap. It is valuable for monitoring plant health, studying plant physiology, and conducting ecology research.
In conclusion, paper chromatography is a simple yet powerful technique for qualitative analysis such as glucose detection. Although it is not suitable for precise clinical measurement, its core principle remain relevant across various industries. In the pharmaceutical industry, it supports quality control of liquid formulation. In the food and beverage industry, it helps identify hidden sugar. In forensic science, it contributes to preliminary biological screening. The experiment demonstrates the use of RF value to identify compound and method similar industrial practice. Future improvement may include use of paper-based microfluidic device to adoption of environmental friendly solvent. Third, integration of digital automation. Advancement enhance industrial relevance and help prepare graduates with essential analytical skill value in today's workforce. This is our references that related.
Thank you. That's all from us. As the same goes, one thing that you can fake is chemistry.